Hey guys, welcome back. Today we are going to be doing a dihybrid Punnett square. So in the last video we learned how to do a monohybrid square. Today we're going to be looking at a dihybrid square. So remember that the prefix di means two, and in this case, because we're doing dihybrid genetics, we are looking at two genes at the same time. So instead of just having our typical two alleles for our one gene, we're actually going to be smashing them together to create one gene here and one gene here. So 1 and 2. These represent our genes. We will have four alleles. Remember, each one of these is an allele. Okay, and we're going to have two genes. Okay, so I'm just going to kind of create like a basic example and do the full thing out for you so you know how to do that on a test. You probably won't have to do the entire thing, but this is just so you know how to do it. So I'm going to give you the cross statement. Okay, I'm going to give you the cross here. Okay, so this is mom and dad. These are the parents. We are, like I said, we are doing two genes. So let's say that this is mom and dad. The parental cross. Okay, so what we're going to do first in this situation is we're all going to agree that we have four letters. We have four alleles present in each parent. Okay, now on this little dihybrid box here, you probably want to say, oh, there's 1, 2, 3, 4 boxes. I'm going to put one of the letters across each one. That is incorrect. Okay, you have to do something called FOIL. Okay, this is the same thing as it was in math. So you're going to do like first, outside, inside, last. So I don't like that because I'm not like a super math inclined kind of person. I like to just think about it like this mentally, but then I'm going to show you like the way that I know how to do it that you will not mess up if you can just remember it. So essentially A is going to pair with B, then A will pair with B, then this A will pair with this B, and this A will pair with this B. And you'll do the same thing for both parent, okay? But I like to just kind of remember it as a formula because I think that includes less opportunity to mess it up. So basically, you're going to remember 13, 14, 23, 24. Okay? What do I mean by this? I mean position number 1, which is A, and position number 3, which is big B. Okay, they are going to pair together just like I drew right here. That was really bad, me following that line, okay? That's what I drew there, A with B, so 1 and 3. Now I'm going to do position number 1 again with position number 4. So big A, little b. So again, that's the same arrow that I drew here. It's 1 with 4. Okay, now I'm going to do the same thing with 2. So I'm going to have 2 with little a and 3 is a big B. That's what I drew right here. And then we have little a with a little b, and that is what I drew right here. Okay, so what we just created right here, okay, these are basically the gametes. Okay, so this is half of the equation. These are the gametes. So if this is mom, each one of these little combinations of genes here could be represented in an egg cell. Okay, we're going to do the same thing over here with dad. So again, we're going to write 13, 14, 23, 24. Now for my kiddos, I really want you to write the numbers out because this provides less opportunity for error and we want to get as many things correctly as possible. Okay, so I need you to write out the numbers 13, 14, 23, 24. And again, all that means is that number 1, which is big A, and number 3, big B, are going to pair together. Then I need big A with position 4, little b, which is right here. Now we're going to do the little a with a big B right here. And then we have little a with a little b, 2 with 4. Okay, that's the same thing as FOIL. Okay, but I really like to teach it with this 13, 14, 23, 24. Okay, yes, you notice that both parents have the same genotype. I picked the hardest one, or quote, the hardest but longest one to show you. That way all the other ones that you ever do will be easier than this, or equivalent at worst, okay? So if this is dad, each one of these could represent the genes that's carried in sperm cells. So you know, in fertilization, when a sperm cell and an egg cell meet up, okay, when they come together, that's going to be fertilization. Okay, and when we recombine those, that's basically this resulting little like miracle child baby, right? That's going to be what we're predicting in this dihybrid square, okay? So now that we have taken the eggs and we have taken the sperms and we have we have taken the sperm and we have discovered what the potential genetic crosses could be, we are now going to put those onto the actual square itself, okay? So we're going to put mom across the top. Doesn't matter if it's across the top or down the side. No, it doesn't, okay? But I'm going to put mom across the top. So this is a little different because now we're going to have 2 letters on each side of the square. And that's important because at the end we want to have 4 letters or 4 alleles total, right? We need 4 alleles total so we can have the correct genetic makeup here. So dad is going to be the same, and I'm just going to put them down the side. It does not matter which way you put it, like in regard to across the top or down the left-hand side. It will be the same, okay? But that's where I put the information right here, this 13, 14, 23, 24. I just added it to our chart, and I added it to our chart, okay? So now all we have to do is go through and fill out the chart. Now, A's touch A's and B's touch B's, always. None of this AB business. A's touch A's and B's touch B's, always. That's how it has to be, okay? So let me give you an example. In this first box, we're going to be dealing with these guys and these. So A and A, B and B. Okay? Now I'm going to go and I'm going to do this one. And this one's going to come over here, okay? So I have A with A, and I have B with B, okay? In the next box, I'm going to have A with A, B and B, A with A, B and B. So I'm going to go through and I'm going to fill this out. If you're following along, try to do this on your own without just copying it to make sure that you can track all of the genetics here to make sure that you fill these out correctly. Okay, yay, done. Okay. So these are the probabilities here, okay? So the next thing that we need to do is the genotype and the phenotype. We're going to do the genotype first. Remember that the genotype is referring to the letters, and the phenotype is referring to the way that it looks, okay? This is a little bit more complicated this time, but imagine that this box is like 1, 2, 3, 4. I'll just number them for the sake of explaining how I do these. I like to take a very logical approach. That way you don't forget any of these little boxes, okay? They should always add up to 16. I'm going to do fractions instead of percentages in this case, just to keep it easy. So the genotype is referring to the letters, and we always start in box number 1. So in box number 1, I see these letters, this genotype. So I'm going to say that I see big A, big B, big B. Okay? How many squares have this exact genotype? If you go through and you look at every single one of them, I see that I have 1 out of 16. Okay? Now I'm going to move on to box number 2 because I finished box number 1. In box number 2, I see big A, big B, little b. That's what I see right here. I also see it right here. I see 2 out of 16. Yes, you can reduce this, but if we're trying to add up to 16, it's easier if you don't. Okay. In box number 3, I see a big A, a little a, big B, right here. I also see that right here. Okay, I see 2 out of 16. In box number 4, I see big A, big B, little b. And here I actually see 1, 2, 3, 4. 4 out of 16. Yes, that reduces, but leave it out of 16. Okay, so we're done with the first row, so I'm going to scroll up a little bit. I also need more room to write. We've already done box 5. We're working on box 6 now. That's why I like to underline or cross out or something. That way I know, put something in the box. That way I know that I've already counted it. Okay, so big A, little b. I only see 1 of those. And I'm good until I get to box number 8, which is big A, little b. And I see 1 of those right here too. So I see 2 out of 16. Okay, I've done 9 and 10. Let's go to 11. In 11, I see little a, big B, right here. And I only see 1 of those. 1 out of 16. In 12, I see little a, big B. Okay, and I also see another one right there, so that's going to be 2 out of 16. And in my very, very last box, I have all of my little letters, my double homozygous recessive. This is going to be 1 out of 16. So all of my boxes have been accounted for. I have underlined something in all of them, which means that I have counted it. So I'm going to go through, and this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So this is 16 out of 16. I have accounted for all of my genes in my genotype, all the combinations, okay? Next, we're going to talk about our phenotype, okay? So I didn't tell you what it's going to look like. So let's just say that I'm just going to define my alleles over here. Okay, I'm just going to say that this is going to be tall and this is going to be short. And we'll say this one is purple and we'll say this one is white, like the Mendel's pea plants. Okay, so the phenotype. I'm going to go through, and just to make this easy on myself, I'm going to highlight all of my capital A's. Okay, now I'm going to do the same thing with all my capital B's. Remember, it only has to be at least 1, right? At least 1 per box, because as long as you have at least 1 capital letter, it's going to show the dominant value, or the dominant trait, rather. Okay. Cool. So here we go. I think that I've got everything on here. Cool. Okay, we are going to start off with our combinations. So our combinations, if we're looking over here, okay, we can be tall and have purple flowers. We can be tall and have white flowers. Or we can be short and have purple flowers, or we can be short and have white flowers. Those are the only combinations. So if tall is dominant, to get a tall plant, I'm looking for an A, and who cares what's next to it, right? It doesn't matter what that second position is. In order to get purple, I'm looking to get a dominant trait. Who cares? So I am looking for something that is A, who cares, B, who cares, just like that. To get tall and white, I need A, who cares, and then I need white is recessive, little b. So I'm looking for A, who cares, little b. Okay. For short, that is recessive. So it has to be 2 little a's. And for purple, it's dominant. So I'm going to have little a, big B, who cares? Because it's dominant. And this one is special. It's going to be all of my little letters. So this one has to be this genotype that we're looking for, okay? So now we're going to take to the square, okay? I am looking for a capital A with a blank, doesn't matter. So a capital A and a capital B. Okay, so I see capital A, capital B, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, I see 9 of them. 9 out of 16. And that's important, so I'm just going to put a box around it. That's a little bit big. Oh, that's how it was. It doesn't want to be delicate. That's okay. Okay, so that one is 9 out of 16. Do you see how it helps when you use different colors? Because like I just looked for the boxes that had both yellow and green in them, okay? Now I'm looking for tall and white, so I'm looking for A with something next to it, but who cares? And then a little b. So I'm looking for an A with 2 little b's. Capital A, I see 1, 2, 3. I see 3. 3 out of 16. Okay. Next, I'm looking for short and purple, which is going to be little a with a big B and something else. Little a, big B, something else. 1, 2, 3. I see 3 out of 16. Okay. 3 out of 16. Okay. And lastly, all of my little letters. I count 1. 1 out of 16. 1 out of 16. Okay, now let's check. 9 plus 10, 11, 12, right? 13, 14, 15, 16. So I have 16 out of 16 means I am done, okay? So this is what one dihybrid question looks like. This is how you would solve it from start to finish. You would do FOIL up here to find the gametes for mom and dad, and the gametes, you remember when they come together, this is what we are predicting. We are predicting that there is a 9 out of 16 chance that these two plants, when bred together, are going to have a tall and purple offspring, okay? So this is a special ratio called the 9-3-3-1 ratio, and that is only when we have a double heterozygous cross. That is the only time that you will have that, okay? So this is how you set up your dihybrid genetics. This is how you do the genotype and the phenotype. It takes a little bit of time, but this is not hard. It's just a little tedious. Be methodical, be logical. If it helps you, I numbered first. I went through and I numbered all of my boxes. I also use different colors to highlight the dominant traits to help me track them a little easier. And in the phenotype, it really, really helps when you go through and you list out all of the options first, because then you know what you're looking for. So all I did to do that is I said, I have tall, it can't go with short because those are two of the same, it's the same gene. All right, we're looking for different genes. So tall and purple or tall and white. Then I did short and purple and short and white. And that's exactly the same FOIL that we did up here, right? I just wrote out the words, and then that's exactly what I looked for when I went through and I looked at my chart to see how many of each of these do I have. Very helpful to go through and use highlighters, especially if you have the ability to do this on paper, or if you don't, I'm just using an iPad to record this, so I just easily just went through. Even if you don't have like the pencil, you can use your finger to just highlight everything, okay? Makes a really big difference when you're learning how to do these, just to help yourself track. Like I said, this is the most complicated. This is the most amount of genotypes that you would have for this specific kind of cross. All the others will have a redundancy and make them very easy, okay? So if you are one of my kiddos, make sure that you have this exact diagram, this work added to your notebook for the class Everyone else, I hope that this was at least a little bit helpful to help you figure out how to solve these dihybrid questions. Thanks, and have a great day.